All right, so we have talked about enzymes, we've talked about what metabolism is, and what I want to talk about in this lecture in particular is because we've addressed cellular respiration, I want to talk specifically now about photosynthesis. So photosynthesis uses a lot of the same principles that we've already discussed whenever we talked about cellular respiration. And we talked about the different ways that you can generate ATP. And we know that through cellular respiration, we see ATP generated via substrate level phosphorylation and oxidative phosphorylation. So through the process of cellular respiration, we do get ATP generation. We also break down organic molecules with the help of oxygen and produce the byproducts, carbon dioxide. So that oxygen is used to bind up the carbons that are released from breaking down the organic molecule, which is why we get carbon dioxide. And then remember, the water is generated because when we get done with all those electrons in the electron transport chain, when we get done with hydrogens, we bind up the hydrogens and the electrons, also with oxygen as a final electron acceptor to create water. So we have another, what we're going to discuss today is the flip side of kind of the ecological metabolic pathway. And we're going to talk about photosynthesis. So it's worth noting that photosynthesis actually uses the products of cellular respiration, that is carbon dioxide and water, in the synthesis of organic molecules. So we're taking 6 carbons in the form of carbon dioxide, 6 carbon dioxides, and we're going to build bonds between those carbons to create 6 carbon glucose. So basically, you hear that this is the opposite. The reactions are opposite, again, because the products and reactants are the opposite. But the actual processes are not that similar, how we get there. So I do want to mention that this does require energy because we are going to build bonds between carbons, and we have to get that energy from somewhere. And in the case of photosynthesis, photo meaning light, that tells you that the energy for this process comes from light. All right, so let's talk about it. So the overall goal of photosynthesis is we want to collect energy from the sun, and we want to use this energy to kind of like release electrons. So in terms of cellular respiration, we had bonds between individual carbons and glucose. We broke those bonds. We didn't talk about how we were able to break those bonds, but we used enzymes to do that. So you have glycolytic enzymes that break apart the bonds in glucose and release those electrons. In photosynthesis, we need to break a bond to release electrons as well. But instead of using enzymes to break that bond, we actually use light from the sun. So with photosynthesis, what we're going to do is we're going to collect energy from the sun to release and energize some electrons. And then just like we did in cellular respiration, at least with oxidative phosphorylation, we're going to kind of carry those electrons to a membrane where we're going to generate a proton motive force, which is going to allow us to generate ATP. And then we're going to use that ATP as well as the electrons we carry to basically build bonds between 6 carbon dioxide molecules. We're going to release the oxygen off those carbons and build bonds between those carbons, eventually creating a 6-carbon compound, glucose. So the overall reaction, like I mentioned, is the opposite of cellular respiration. We use 6 carbon dioxides, 2 molecules of water, as well as some light. And our products are going to be a 6-carbon organic molecule and oxygen. So photosynthesis occurs in 2 distinct steps. You have the light-dependent step. This is called the photosystems. So there's actually 2 photosystems. We'll talk about this. The photosystems are used to generate our ATP as well as to harvest electrons. So this is where we get the energy and the electrons that we're going to use in the second step, which is the light independent step, or the Calvin-Benson cycle. And the Calvin-Benson cycle requires quite a bit of energy and quite a bit of electrons because it's going to be building bonds between carbons to generate glucose. The product of the Calvin-Benson cycle is oxygen. Because we're using carbon dioxide, we're going to use that carbon to create glucose, but obviously that means we're going to be freely releasing that oxygen. All right, so let's talk about it individually. So first off, where does this occur? We know that cellular respiration occurs, glycolysis occurs in the cytoplasm, whereas the last three steps, pyruvate, oxidation, Krebs cycle, and electron transport chain takes place in the mitochondria. And the reason it takes place in the mitochondria is because we need that membrane for oxidative phosphorylation. Photosynthesis, because we also, as we mentioned, generate or utilize an electron transport chain, have to generate a proton motive force to generate some ATP, we also need a membrane. So photosynthesis uses a chloroplast. Chloroplasts contain or kind of have two components, just like the rest of the cell, just like the cytoplasm, and just like the mitochondria, which has a mitochondrial matrix. A chloroplast does have sort of like a jelly-like interior, and this is called the stroma. So this is sort of like the fluid or jelly-like stuff inside of this sac. Okay. And then you also have lots and lots and lots and lots and lots and lots and lots, just like a mitochondria, lots and lots and lots and lots and lots of folds of membrane, which are really important because, again, we need a membrane to generate a proton motive force. Those membranes make up what are called thylakoids. So outside of these thylakoids, we have the stroma, and then the thylakoids are made of membrane, and then inside of the thylakoids, we have some more of that kind of like jelly-like material, which is called the thylakoid space. So you'll notice that in this picture, the thylakoids are green. That's because the thylakoids, those membranes, are also where we have pigment stored. So pigment is really important because it is pigment that absorbs the light from the sun. That's what allows us to absorb light from the sun and utilize energy in the form of photons, basically in place of an enzyme. So the light reactions, because they need that pigment and because they need a membrane, the light reactions occur in the thylakoids. However, the dark reactions do not need membrane. They just need all the energy that's generated in the thylakoids. So the dark reactions actually occur in the stroma of a chloroplast. All right. So again, this picture on the right kind of shows an overall summary of photosynthesis. You have the light reactions, which occur in the thylakoids. What's happening here is you're going to have water and light enter the thylakoids. That light actually breaks apart water. And what is released from that water is we're breaking a bond. So we have electrons released. And we use those electrons, we bind up those electrons with an electron carrier, NADP, to create NADPH, which actually carries 2 electrons. And then we also utilize electrons to generate some ATP. So the light reactions generate energy in the form of ATP. They also release electrons, which will be able to be used in the Calvin cycle. We do release some oxygen during the light reactions because we're splitting water. So we have to use the hydrogens to generate a proton motive force, but the oxygen we can actually freely release as oxygen. We do also get oxygen released in the Calvin cycle because we have carbon dioxide put in, and we will release some oxygen through that process as well. So it's important to note that the energy and the electrons generated via the light reactions are used to build bonds between carbon molecules in the Calvin cycle to create sugar. So that's the overall reaction. So let's start with the photosystems. The photosystems can be broken into two parts. Oh, well, this is just a summary, so never mind. I got a little bit ahead of myself. So this is a summary of photosynthesis in general, kind of what I mentioned on the previous slide. You have the light-dependent steps. These are the photosystems. There's two photosystems, Photosystem II and Photosystem I. Photosystem II actually occurs first. It's named 2 and 1 just kind of in the order it was discovered. But Photosystem II occurs first, and what happens in Photosystem II is that light is absorbed, and this splits water, releases electrons. This will generate a proton motive force along with hydrogens, very similar to what we see with the electron transport chain in cellular respiration. And then that electron transport chain will be utilized to create ATP. So Photosystem II leads to the production of ATP. Photosystem I occurs next, and what happens is we use those electrons, which were already used in the electron transport chain, but now, instead of using oxygen as a final electron acceptor, what we use as a final electron acceptor is NADP. And NADP grabs those electrons, it's reduced, so it becomes NADPH, and then we're going to be able to use that NADPH to kind of contribute electrons to the anabolic reaction in the dark, in the Calvin-Benson cycle. So the overall purpose of the light-dependent photosystem II is to create ATP, and photosystem I is to kind of gather up electrons that we're going to use to build bonds in the next step. This is called light-dependent because you have to have that pigment absorb the light in order to split water. So this process, the photosystem, can only occur when you do have light. And it generates all the energy. Then you have the Calvin-Benson cycle, which is also referred to as the light-independent step. So this is really important to note that even though this is called the light-independent step, that doesn't mean that this reaction can necessarily occur in the dark. It is, even though it directly does not require light because it doesn't have any involvement of pigment, it is dependent on the energy that is generated during the photosystems. So the Calvin-Benson cycle, at least in traditional photosynthesis, the Calvin-Benson cycle cannot occur without the photosystems occurring. So even it's called the light independent step, not because it doesn't require light at all, but it's called light independent because it doesn't involve any direct absorption of light. There's no pigment involved, right? So it's using energy that's generated during the light dependent step. So again, during the light dependent step 1, you have the photosystems. The photosystem II generates ATP. Photosystem I harnesses electrons and reduces NADP to NADPH. And then that ATP and those electrons are carried to the Calvin-Benson cycle. And it's that energy and those electrons that fuel the Calvin cycle. And that energy is used to basically create, so remember biosynthesis, is creation of biological molecules. What we're using is that energy in order to build bonds between these carbons. And so if you have 6 carbon dioxide, you can build a 6-carbon glucose. It's a little more complicated than that, but that's kind of like the essence of it. 6 carbon dioxides are used to generate 1 glucose. All right. So this is looking, what I want to do is kind of go through each of these individually. So we have, first, photosystem II occurs. The slide's a little crazy, but we can go through it. So what you're looking at is the thylakoid membrane. So the inside of that thylakoid membrane is thylakoid space, and then outside of that you have the stroma. And you can see what happens is light is absorbed by the green pigments. And what that means, what happens then is that basically those photons energize electrons found in those pigments. And so you can see those two little tiny electrons. What happens is all of this energy is used to break water apart. So H2O is split into H and oxygen, but also from that bond breaking, that's where we get those electrons. So yes, the light is used to split water, but also make sure that those electrons are released and very energetically charged. So the first step of Photosystem II is that light is absorbed from the sun, and this light basically allows water to break, which allows us to release electrons. This releases oxygen, which is why we get oxygen as a product of photosynthesis, but also this releases 2 electrons, and you can see 2 hydrogen ions. So then what happens is those electrons and hydrogens, you can kind of see them moving over here. Notice we see an enzyme that's very similar to what we saw during the electron transport chain and cellular respiration. What happens is hydrogens are going to build up in this thylakoid space compared to the amount of hydrogens outside in the stroma. So you get a concentration gradient, a very high quantity of hydrogens in the thylakoid space compared to the quantity of hydrogens in the stroma. So again, just like in the electron transport chain of cellular respiration, across the thylakoid membrane, you have a proton motive force. So again, that's like the Hoover Dam. You can think about all the water being stored in the thylakoid space, and that fuels ATP synthase, is basically like opening the dam doors. So you can see hydrogen shooting through. Every 1 hydrogen leads to 1 ATP. So that tells us that we can basically get 2 ATP for every water molecule, because every water molecule gives us 2 hydrogens, and that will give us, again, giving us 2 ATP. So that's Photosystem II. Photosystem II, we absorb light. That light breaks water, releases electrons and hydrogen. Basically, those electrons, we'll talk about what we do with those electrons, but one thing that those electrons can help us do is make sure that there is a high concentration of hydrogens in this thylakoid space. So we have that concentration gradient that allows ATP synthase to use those hydrogens to generate ATP. So Photosystem II, we get ATP generation because of water being split. And then, so we've generated ATP, and now we have those electrons. So now what we need to do is use those electrons because we need those electrons to build bonds in the Calvin-Benson cycle. So what we do, so what I did is I kind of like took this screenshot from here. This is all linked. Is we have Photosystem I. So we still do have light absorbed during Photosystem I, but what light does here is it basically allows these electrons, it basically gives enough energy to form a bond between NADP and a hydrogen, basically accepting those electrons. You can see little indentations. You see the electrons. Just like we saw with NAD and FADH2 with cellular respiration, NADP is reduced. And so it accepts those electrons. Also, it accepts a hydrogen because every time you accept electron, you also have to accept some hydrogens to neutralize it. So what happens in Photosystem II is we absorb light from the sun, split water, generate ATP via proton motive force, electron transport chain. And then during Photosystem I, we basically grab those electrons that were released from water and reduce NADP into NADPH. At this point, you can see both ATP and NADPH go to the Calvin cycle. All right, so what do they do in the Calvin cycle? Well, this is just looking at the entire process. So you can see Photosystem II and Photosystem I on that thylakoid membrane. Photosystem II generating ATP, Photosystem I reducing NADP into NADPH, both of those molecules, NADPH, will be the source of electrons to build chemical bonds. ATP will be the source of cellular energy to build chemical bonds. So both of those are moving into the Calvin-Benson cycle. All right, so this is just again a summary of those two processes. Photosystem II led to the generation of ATP. Photosystem I reduced NADP into NADPH. So we have energy and electrons going into the Calvin-Benson cycle. Energy and electrons, those are two things you need to build chemical bonds. So we are ready. We have everything we need to start building bonds between carbons, generating a sugar. Okay, so we have the energy, we have the electrons. Now, what we need to do is, again, complete what's called carbon fixation. So I really want to emphasize here that this is an extremely important process because very rarely in nature do we have the ability to create organic molecules or create sugars. So the dark reactions, the Calvin cycle, one of the kind of biggest things in nature that is difficult to do is to convert inorganic molecules into organic molecules. So that's a very challenging thing to do. The Calvin cycle is one reaction that exists that allows us to do this. And so how it does this is it uses a lot of ATP. And also we have to have a lot of those electrons available to build those bonds. Both of those were obtained through the photosystems. So we have both ATP and electrons. And now what we need to do is take carbon dioxide, which is an organic compound, split it, and then use those carbons to build a sugar. So this is a cycle, very similar to how the Krebs cycle was a cycle, and it has a recyclable organic compound that allows us to go from inorganic to organic compound. So this recyclable compound that allows us to do that, very importantly, is a compound called it's a 5-carbon compound, and it's called ribulose bisphosphate, or for short, the way you'll hear it always, is RUBP, ribulose bisphosphate. What this does is this 5-carbon organic compound binds one of the carbons from the inorganic carbon dioxide, and that binding converts this inorganic carbon into an organic carbon compound. So this is where we have, again, carbon fixation means we're taking inorganic carbon and fixing it. And fixing it means it's like basically sticking it in bond with other molecule, fixing it into an organic compound. This occurs 3 times to generate an organic compound called glucose 3-phosphate, and then you have 3 carbon-containing compound, you have a 3-carbon containing compound, and basically you bind 2 of those together and you get a 6-carbon glucose. Importantly, just like we talked about with oxaloacetate in the Krebs cycle, RUBP is regenerated with each turn of the Krebs cycle, or with each turn of the Calvin cycle. So you do not need to memorize, just one second. Sorry, just one second. You do not need to memorize this entire cycle, okay? What I want you to know, just a second. Thank you. Awesome, thank you. Sorry. What I want you to know is a couple of things. So as you can see at the top of the screen, I mentioned that the output of the Calvin cycle is actually not a glucose, a 6-carbon glucose. The output of the Calvin cycle is actually a 3-carbon molecule called glucose 3-phosphate. Glucose 3-phosphate. So it's actually, again, a 3-carbon molecule. So what happens with the Calvin cycle, because we're just generating a 3-carbon molecule, we actually just need 3 carbons to enter the cycle. So what happens, in here, you can see that we have our RUBP, so our 5-carbon recyclable ribulose bisphosphate, and we have an enzyme that basically allows our 3 carbon dioxides to bind to RUBP, and this enzyme is called Rubisco. So I do want you to remember Rubisco is the enzyme that catalyzes the basically incorporation of carbon dioxide with RUBP, and then this forms a 6-carbon compound called PGA. So again, we're not going to get into the details of this, but I do want you to know that the molecule that kind of allows the cycle to continue is RUBP, and the enzyme that allows the binding, the initial binding of carbon dioxide to the RUBP is called Rubisco. So that is carbon fixation, is when we have that carbon dioxide affixed to that RUBP. And what happens again is it actually takes 3 RUBP molecules and 3 carbon dioxides. You get 6 of these PGAs. A couple of other things take place, but basically you have 3 cycles of this, and through those 3 cycles, you generate 1 glucose 3-phosphate. So that means that 3 turns of this cycle gives you a 3-carbon organic compound, that glucose 3-phosphate. So what that means is you would have to do that twice, which means a total of 6 cycles to generate 2 glucose 3-phosphates, 2 3-carbon compounds, stick those together to generate a 6-carbon compound. Again, we're not going to go through the details of this process. We don't need to know the details of this process, but I do want you to know that you do have to do 6 cycles of the Calvin cycle in order to generate one glucose. And so again, this requires carbon dioxide as an input, it requires our recyclable RUBP, and it requires the enzyme Rubisco for carbon fixation. All right. So again, you do not need to know the details of that, but I do want you to know that the enzyme Rubisco is what helps that process take place. Okay, so this is just a slide showing a summary of photosynthesis. We really did just glaze over it, but even though we learned kind of like the basic steps of cellular respiration and the basic steps of photosynthesis, that will really give you a good kind of foundational understanding of if you go into classes that kind of expand on these processes. So there are a lot more steps to these processes, but we just don't need to know them for this class. You'll have classes that will elaborate on these details. I think my class, molecular biology, when I took it, was literally like a semester of learning cellular respiration. So you will learn these processes in a lot more depth. That's just not the purposes of this class. We're just learning the basics. So like I said, with cellular respiration, I want you to know the basics of what occurs during the 4 steps: glycolysis, intermediate, Krebs, and the electron transport chain, the energy that's generated. With photosynthesis, what I want you to know is the basics of photosynthesis. So when I'm talking about the basics, I'm talking about knowing the purpose of Photosystem II, so that light is absorbed to split water, release electrons, and hydrogen. Both of those are used to generate a proton motive force to generate ATP, but then those electrons are also used to reduce NADPH. And both that ATP and NADPH are used in the Calvin cycle. So you can see when we use ATP here and when we use that NADPH, but essentially they're just used as a source of electrons and energy to allow the Calvin cycle to take place. And the Calvin cycle is when we use carbon dioxide in combination with Calvin cycle recyclable compounds, RUBP, and the enzyme Rubisco, to basically affix this inorganic carbon source, carbon dioxide, into an organic carbon source. So again, 6 cycles of the Calvin cycle have to take place to allow you to generate a 6-carbon compound glucose. It is a very energetic, it requires a lot of input of energy to do this. This is just a summary of these processes and how these are linked. Again, the light reactions, Photosystem II and I, are what generate our energy, and then the Calvin cycle, or the dark reaction, is what uses that energy to generate our organic compound. And again, I just want to reiterate that cellular respiration and photosynthesis are quite linked. Cellular respiration basically uses organic molecules and oxygen to generate energy. The byproducts of that are carbon dioxide and water, which are actually the reactants of photosynthesis, which uses energy from the sun to generate potential energy in organic molecules, which is then converted into kinetic energy through cellular respiration, and so on and so on and so forth. These two metabolic systems are very intimately linked because they share products and reactants. I do want to note a common misconception, and I think we've already addressed this in class, but cellular respiration generates ATP. Photosynthesis generates a little bit of ATP, but uses that ATP in the Calvin cycle. So photosynthesis overall is a very endergonic process, a very energy-consuming process. Cellular respiration is really good at generating energy or releasing free energy. So in plants, where you have a chloroplast, just because you have a chloroplast does not mean you don't have a mitochondria. So plants actually have to do both of these. Plants have both chloroplasts and mitochondria, because plants have to absorb light from the sun to generate their sugar, and then can use that sugar to create ATP via cellular respiration. So plants have a really huge advantage here, right? Because they can be very independent. So if you have a chloroplast and a mitochondria, what that means is you can create your own organic compounds, your own sugar. And then once you create sugar, you can metabolize it into ATP, into energy. Whereas humans, or any organism that does not have the capability of photosynthesis, we actually have to consume our nutrient sources. So I have to get my sugar from eating plants or eating things that ate plants. And that's how we get our sugars. We can't make our own. We have to consume it. So that's another difference between producers. Well, that is actually the primary difference between producers and consumers, is that producers in an ecosystem can actually produce as well as consume their own organic molecules, whereas consumers have to consume producers to get those organic molecules. But plants can do both, so they're super independent. This is just kind of talking about that cycle and consumers and producers that I just talked about, but I'm not going to go too much into this. And that's the end of this lecture. If you have any questions, please, please, please reach out. Put them in the discussion board so that we can collectively figure out what the questions that are overarching in the class are, and then I can make another supplemental lecture if we need. All right.